Good morning or good afternoon. So my name is Claudia, and my title is One Disease, Too Many Genes, One Gene, Too Many Diseases. So this is a provocative way to introduce something that is called complex neurodegenerative disease. So complex neurodegenerative diseases is actually a group of diseases. Some of them are famous because they get a lot of coverage in the media. For example, Alzheimer disease or Parkinson disease. But there are many other complex neurodegenerative disorders that actually are not that famous because they are less frequent in the general population. So they don't get taught that much. For example, we have dementia with Lewy body, which is an in-between Alzheimer and Parkinson. It's a form of dementia, so we have the cognitive impairment, but we also have the motor component that is typical of Parkinson. And then we have frontotemporal dementia, which is the second most common form of dementia after Alzheimer's disease. And then we have peak disease, progressive supranuclear palsy, and so on and so on. Unfortunately, this list is very long. There are a lot of disorders that can be classified as complex neurodegenerative diseases. But complex neurodegenerative disease is a mouthful. So let's try to break it down. Neuro. Because these disorders are all due to the loss, to the death of brain cells, which are called neurons. Degenerative. Because these disorders are progressive. So they start mildly and they progress over time. And the progression is caused by the fact that there is an ongoing brain degeneration. So the neurons, the cells in the brain die and they cannot be replaced. So there is this progression of degeneration of the brain tissue that cannot be healed. And then complex. And this is the point of this talk today. These disorders are complex. It means that there is not just one simple causative event that triggers the disorder, but there are multiple factors. Some of them are genetics, some of them are from the environment, and it is the mix of these factors that is actually the cause of this disease. So let's go a little bit into the genetics. So the complex neurodegenerative disorders, they come in two different groups. Some of them are familial, some of them are sporadic. The tip of the iceberg, so the minority of the cases are familial. This means that the disorder is passed down from generation to generation, is inherited within the family. The majority of cases actually are sporadic. So there is no positive familial history. This is telling us something about the genetics. And in particular, it's telling us that in the familial forms, we have a very strong genetic component at the base, at the cause of the disorder, while in the sporadic forms that are not inherited in the family, there is a weak genetic component. But what is a genetic component? So the DNA is a vast instruction manual that we have in every one of each cells in our body. It is a sort of library, and we call this library genome. So the library is made up of books, while the genome, our library, is made up of genes. And the books, so the genes, are actually they are containing all the recipes for the cell to function. So let's look at one example. The cell has to carry on a lot of function to stay alive. And one of the functions that is required for the cell to survive is to dispose of its own waste. And actually, it's called autophagy. Is a Greek term that means self-eating, because the cell is disposing, is recycling its own waste. So it's like if the cell is eating itself. This is a complex function that the cell has to carry on. And we can break it down into smaller steps. So first, the cell has to have the information in order to detect the waste. So there should be a collection of books or genes in which we have stored all the information for the cell to detect the waste. And then we need another collection of books or genes with all the information for recognizing the previously detected waste. And then we need information on how to gather the waste and to send the waste to the recycling facility. And finally, we need all the information for running the recycling facility. And this is just one function, the cell waste disposal. Imagine there are a lot of other functions that the cell has to carry on in order to survive. In fact, in our DNA library, in our genome, we have around 20,000 books, so 20,000 genes that are storing the information. So let's go back to the cell waste disposal. And let's look at this function at this step in which the cell is basically gathering the waste and sending the waste to the recycling facility. What happens if there is a mistake in one of the books or one of the genes? So the information that is stored in one of the books is wrong. We call this situation a mutation. This is a problem because the cell has the wrong information. So the cell probably is not going to be able to gather the waste and send it to the recycling facility very well because the information on how to do that function is wrong. When we have a situation like this, a mutation, we very likely go to have a disease because the function is impaired. But there is also another condition, another situation that we have to discuss, and is the inter-individual variability. So the books are not written in the same way into different people. We are all different. This is called variation, genetic variation. So the way that books, that my books are written is different from the way that your books are written. So this means that if we have subject 1 and subject 2, maybe the books in subject 1 are written slightly better than the books in subject 2. This is not causing any damage. The cell is still able to go on with the function. However, subject number 1, it will be able to carry on that function better than subject 2 because the book is written in a better way. This situation is called risk factor. So if the book is written badly, then the cell is carrying on that function slightly in a worse way in comparison with like the general population. So if we go back to the genetic image, we have the familial forms in which we have a very strong genetic component. So the disease is passed down through generations because we have a mutation in one of the genes, in one of the books. So there is a strong genetic component that is causing the disease to happen. While in sporadic forms, we don't have a mutation, we have a weak genetic component, we have genetic variability. So some individuals carry variability that increases their chance or risk of getting the disease because one or multiple books are badly written. So if we now look actually at the disorders, let's look at Parkinson. These are the genes that are mutated in familial Parkinson disease. We know around 15, 20 genes, depending on how we classify Parkinson. And if a mutation is present in one of these genes, this is a very strong genetic factor that is predisposing to getting Parkinson. On the other hand, we have sporadic Parkinson. And we know around 70 genes that can be poorly written, and if they are poorly written, they increase the chance of a person to get Parkinson disease. So we have around 20 genes associated with familial Parkinson, around 70 genes associated with sporadic Parkinson. And if we look at frontotemporal dementia, again, we have 10 genes that we know when there is a mutation, this is able to cause frontotemporal dementia in families. And we have 4 genes that we know if they are poorly written, they will be a risk factor. So they will increase the chance of this person to get frontotemporal dementia in their life. So as you can see, there are a lot of genes associated with a single disorder. This is why the title is One Disease, Too Many Genes, because there are many genes that are associated with the same complex neurodegenerative conditions through mutations in familial forms or as risk factors for sporadic forms. But there are a lot of genes. What about the second part of the title? So one gene, too many diseases. And let's take the example of frontotemporal dementia and amyotrophic lateral sclerosis. So frontotemporal dementia, as said before, is a form of dementia, so there is cognitive impairment, while amyotrophic lateral sclerosis is a motor disease, so the motor function is affected first. Then we can have dementia, but the first thing is actually the alteration of the movement. So these, from a clinical point of view, these are very different clinical presentations. However, if we look at the genetics, we have in blue 3 genes that we know are only associated with frontotemporal dementia. In yellow, we have 5 genes that are only associated with amyotrophic lateral sclerosis. But in the middle, you see we have a list of genes that can be associated with both. So if we get a mutation in one of those genes, we can have frontotemporal dementia, so a form of dementia, or we can have amyotrophic lateral sclerosis, so a form of movement disorder. And so it's very confusing. How is this possible that the same gene can be actually associated with 2 different disorders? So what do we think is happening here? Why so many genes, and why one single gene can be shared across diseases? And the answer is basically in the fact that these disorders are complex. So it is unlikely that there is just one simple causative event. It's probably, the triggering event is probably a combination of multiple factors. And in the lab, we have this cocktail theory. So it's not one single ingredient to cause the disease, but it's the concoction of multiple ingredients that is actually able to trigger the disorder. For example, let's think in this case is Parkinson. So we can have synuclein, the first gene in orange, and then we have LRRK2, the second gene in yellow. If we have mutations in this gene, this is a very strong genetic ingredient to get familial Parkinson disease. And then we take a risk factor in blue, which is a gene badly written, which increases our genetic predisposition of getting Parkinson. And then we have another risk factor in pink, which is an environmental factor, so something that we are exposed in the environment that can increase our chance of getting Parkinson. And now let's prepare some cocktails. So if we put together, for example, a strong mutation in LRRK2 plus a gene that is badly written and increase the risk of Parkinson, then we get Parkinson in this person. And this would be a familial case of Parkinson since there is a strong mutation in LRRK2. But then we can also have another patient in which we only have 2 genes that are badly written plus an exposure to something in the environment. And the combination of these 3 things is actually increasing the chance of this person to get Parkinson at the point that this person starts having Parkinson. And finally, we have another person with another strong mutation in synuclein, plus 2 genes badly written, and then we have Parkinson. So you see that these 3 people, all of them have Parkinson, but the reason why they have Parkinson is totally different. And then we have the second part of the title. So one gene, too many diseases. And so now let's take the example of frontotemporal dementia and amyotrophic lateral sclerosis. So we have C9orf72, which is in orange. And this is a gene that when it is mutated, it can give frontotemporal dementia or amyotrophic lateral sclerosis. This is one of the genes that was in the middle of the slide before. And then we have in blue a risk factor, so a gene that is badly written and is increasing the risk of getting frontotemporal dementia. Then in pink, we have an environmental risk factor, so something in the environment that we get exposed to and is increasing our risk for getting amyotrophic lateral sclerosis. And then we have another risk factor in green. In this case, is a gene badly written that is increasing the chance of getting amyotrophic lateral sclerosis. And when we prepare the cocktail, we put in both the two cocktails C9orf72, so it's the same gene with the same mutation. And then in the first cocktail, we add the risk factor for frontotemporal dementia, while in the second cocktail, we add the risk factor for amyotrophic lateral sclerosis plus the environmental exposure that is increasing the risk of getting amyotrophic lateral sclerosis. And you see that even though the same gene, C9orf72, is present in both the two cocktails, one patient will go on developing frontotemporal dementia, while the other one will go on developing amyotrophic lateral sclerosis. And in our lab, what we do is trying to find ways to analyze the cocktails. Because biomedical research up to now has been organized in a way that we study one gene at a time, one gene as a singular entity in isolation. But we don't have the techniques, we don't know how to approach the study of multiple causative events all together. So this is why we are working in that direction, and we think it's very important, and I will give you an example of why we think it's important. Here we have two cocktails. We have granulin, GRN, plus a risk factor. So GRN is a gene that can be mutated in frontotemporal dementia. And then we have that risk factor, which is a gene badly written, which increases the chance of getting frontotemporal dementia. So the first patient has frontotemporal dementia. The second cocktail, multiple risk factors, again, for frontotemporal dementia. So these two patients, they both have frontotemporal dementia. And now we have developed a therapy that is able to target GRN, granulin. Do you think that that therapy will work for both the two patients? We don't know, because the first patient has the mutation in granulin, so we can treat this patient with the drug that we have developed against this strong genetic factor. But what about the second patient in which this gene is not mutated? They have the same disorder, but for different reasons. So it is important to study the cocktails because we want to understand whether therapies will work for all the patients or if we need to stratify patients based on the genetics before giving them a therapy. And the way that we do study the cocktails is through computer science and mathematical modeling. We use networks, which is like the design, the graphics at the very bottom of the slide. And there is a branch of mathematics, which is called graph theory, which is actually devoted to the study of network. And this is what we use to take into consideration so many genes and so many factors all together. And this is the group that I work with. Thank you very much for your attention.